This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to a journey into the deepest mystery of our existence. Traditionally, we've searched for life's origin in a single, lucky molecule. But what if life began as a team effort? Today, we'll explore the collective intelligence of the primeval soup and see how life functions as a planetary force rather than a solitary event. From systems chemistry to the architecture of interdependence, we are shifting our lens from the individual to the group. Let's discover how the universe organizes itself into the complex, interconnected wonder we call life. Chapter 1. The Dawn of Chemical Collective Intelligence. The quest to understand how life began on Earth has traditionally focused on a single transformative moment. We often imagine a lonely, first-of-its-kind molecule or a solitary primitive cell suddenly flickering into existence against a backdrop of sterile rock and water. However, a growing body of scientific thought suggests that this individual-centric view might be missing the most important part of the story. Instead of looking for a single hero molecule, researchers are increasingly exploring the idea that life's origin was a collective effort. This perspective shifts the focus from the individual to the group, proposing that a form of collective intelligence emerged within the primeval chemical soup long before anything we would recognize as alive ever appeared. To understand this, we have to rethink what we mean by intelligence. We usually associate intelligence with brains, nerves, and complex animals. But in the context of the early Earth, theorists are defining it as a specific kind of computational process. In this view, the early oceans were not just a random mixture of chemicals, but a vast natural laboratory where information was being processed. Molecules were not just static bits of matter, they were information carriers. As they moved through the water, driven by the chaotic energy of heat and currents, they were constantly bumping into one another. These interactions were not just random collisions, they were a form of natural inference. When certain molecules interacted, they could calculate a response, such as bonding together or breaking apart, based on their shapes and electrical charges. This environment is often described as a quasi-chaotic system. While it looked like a mess on the surface, there was an underlying logic to the way these molecules behaved. This is where the concept of collective intelligence comes in. A single molecule can't do much on its own, but a social structure of molecules can perform tasks that are impossible for any individual component. Scientists suggest that this elementary chemical social structure was firing its collective intelligence for the sake of survival. In this pre-biotic world, survival didn't mean staying alive in a biological sense. It meant persistence. It meant finding ways to stay together and continue reacting despite a volatile environment. This successful collective action boosted the growth of complexity, eventually leading to the emergence of well-defined life. The raw materials for this collective intelligence were not generic chemicals, but specific feedstock molecules that set the stage for a chemical Big Bang. Researchers have identified hydrogen cyanide, HCN, and hydrogen sulfide, H2S, as the primary precursors in this process. When these molecules are combined with ferrocyanides and exposed to the energy of the early Earth, they don't just react, they explode into variety. This phenomenon was vividly demonstrated in the experiments of Bob Hazen, who used pressure bombs, small high-pressure vessels, and gold capsules to simulate the crushing conditions of the early Earth. Hazen observed that a few simple ingredients, when subjected to these intense conditions, could transform into tens of thousands of different compounds in the span of a single lunch break. This rapid diversification is the first hallmark of a collective system beginning to explore its own potential. We can see the visual signature of such collective behavior in the study of bioworms. These are intricate patterns created by microbial communities that follow simple, local rules of growth and competition. No single microbe is designing the pattern, yet the community as a whole produces a complex, recognizable structure. This is collective intelligence in action, leaving a physical mark on the world. In the prebiotic era, similar rules governed the interactions of molecules. They didn't need a leader or a blueprint. They simply followed the physical and electrical lock and key mechanisms inherent in their structures. Simple organic compounds naturally combined into more complex ones, leading to the emergence of patterns that weren't imposed from the outside, but emerged from the interactions within the group. One of the most powerful ideas in this collective framework is the concept of a hypercycle. Imagine a group of different molecules, where the first molecule helps in the creation of the second helps the third, and the final molecule in the chain facilitates the reproduction of the first. No single molecule in this loop is self-replicating, but the group as a whole is. This is a reinforcing feedback loop, a classic hallmark of collective behavior. It is a strategy where the relationship between the components is more important than the components themselves. In this scenario, life doesn't start with a master molecule. Instead, it starts as a team of simpler molecules that work together to maintain their collective existence. The environment provided specific matchmakers that facilitated this collective assembly. While the open ocean was vast and dilute, certain materials common on the early Earth acted as catalysts for group interaction. Volcanic glass, for example, has been shown to facilitate the assembly of RNA from its component parts. By providing a surface where molecules could be corralled and oriented, these materials forced the chemical soup into a social structure. The glass provided the stage, and the molecules provided the performance, learning to organize themselves so effectively that they eventually moved toward independence. Even the way these early systems managed energy was a collective effort. Every chemical reaction requires an energy threshold to be met. In a collective system, the energy released by one reaction can be used to fuel the next. This creates a metabolic network where the group becomes an expert at harvesting energy from the environment, whether from sunlight, geothermal heat, or chemical gradients. This collective energy management is much more resilient than any individual reaction. If one part of the network fails, the rest can often compensate. This resilience is a key feature of intelligence, allowing the system to maintain its order against the universal tendency toward disorder. We can see the remnants of this collective beginning in our own cells today. The machinery that translates genetic information into proteins is a hybrid made of both RNA and proteins. It is a fossil of a time when these two types of molecules were first learning to work together. Neither can function without the other, and their partnership is the foundation of all modern biology. This suggests that the individual cell we see today is actually the result of a long history of collective interactions. We are not just a collection of parts. We are a collection of partnerships. If collective intelligence emerged before biological life, then the prebiotic period was a lot more active than we previously thought. It wasn't just a long wait for the right molecule to appear. It was a period of intense group experimentation. The first elementary collective intelligence may have emerged in the chemical soup much earlier than the first cell, providing the momentum needed to build life as we know it. In this sense, intelligence didn't just evolve from life, it helped create it. This shift in thinking suggests that the path from chemistry to biology is driven by universal principles of collective organization. We are moving away from the idea that life is an incredibly unlikely chemical accident and toward the idea that it is a natural, emergent property of the universe. If the emergence of life is a group effort, how then do we begin to analyze these complex chemical networks not as isolated reactions, but as entire functioning systems? Chapter 2: The Symphony of Systems Chemistry. For decades, the search for the origin of life followed a path of increasing specialization. Scientists sought to solve the mystery by breaking it down into its smallest possible components, hoping that if they could just understand how a single amino acid formed, or how a lone strand of RNA began to replicate, the entire puzzle would fall into place. This reductionist approach was incredibly productive, giving us a deep catalog of the individual parts that make up a living cell. But it also led to a fragmented field, where researchers often worked in silos, focusing on their specific world, the RNA world, the lipid world, or the metabolism-first world. Today, a profound shift is occurring. The scientific community is moving away from looking at these components in isolation and is instead embracing a holistic systems thinking approach. This perspective suggests that life did not emerge from a single lucky molecule, but from the complex interactions of an entire chemical system, where the relationships between the parts are more important than the parts themselves. To understand this shift, we have to look at what it means to view abiogenesis through the lens of systems chemistry. In this framework, a system is defined as a group of interacting, interrelated, and interdependent parts that form a unified whole. The crucial insight is that the arrangement of these parts is significant. You cannot understand the behavior of the whole by simply looking at the properties of the individual components. Instead, you have to look for emergent properties, behaviors that only appear when the components interact. Life itself is increasingly seen as the ultimate emergent property. It is not a substance you can hold in your hand. It is a process that arises from the way a chemical network organizes itself. One of the most definitive breakthroughs in this holistic approach comes from the work of John Sutherland and his team. For nearly half a century, prebiotic chemistry was haunted by what researchers called the nucleotide problem. Traditional chemistry suggested that to build RNA, one had to first synthesize a sugar, ribose, and a nucleobase separately, and then find a way to couple them together. This proved to be a monumental failure. The conditions required to make the sugar were incompatible with the conditions required to make the base, and even when both were present, they refused to link up in a productive way. Sutherland's breakthrough was to stop trying to build the parts in isolation. Instead of a linear assembly line, he demonstrated a systems synthesis. By starting with a mixture of simple precursors like cyanamide and cyanoacetylene, his team succeeded in building the sugar and the base simultaneously as a single fused system. The components emerged together from a shared chemical pathway, bypassing the need for a difficult coupling step. This experiment proved that the kit of parts for life is not a collection of independent inventions, but the result of chemical contiguity, a single interconnected reaction network where the presence of one molecule facilitates the birth of the next. This systems-level view also changes how we think about the metabolic core of the first living systems. A research team in Germany recently identified a specific set of 402 reactions that likely constituted the core metabolism of the last universal common ancestor, or LUCA. What makes these 402 reactions so significant is that they are self-sustaining and entirely independent of ATP, the complex energy-carrying molecule used by modern cells. This suggests that a robust, functional metabolic network existed long before the arrival of the sophisticated protein machinery we see today. These reactions are exergonic, meaning they release energy as they proceed, allowing the system to drive itself forward using nothing more than the simple gases and minerals available on the early Earth. The role of minerals in this process has also been refined through the lens of systems chemistry. Rather than acting as passive templates, specific metals are now seen as active participants in the chemical circuitry. Ferrous iron, which was incredibly abundant in the early oceans, has been shown to drive 9 out of the 11 steps of the Krebs cycle, the fundamental engine of metabolism. This discovery bridges the gap between inorganic chemistry and biology, showing that the heart of life's energy processing was already beating in the presence of simple iron catalysts. Furthermore, researchers have identified manganese as a vital but often overlooked link. Manganese functions as a chemical bridge between the solid Earth and emerging biological systems, facilitating the transition from mineral-based reactions to the fluid organic networks that would eventually become life. This focus on the collective behavior of chemical networks allows us to move beyond the RNA world or metabolism-first debate. In a systems framework, these are not competing theories, but different facets of the same process. The early Earth was a place of intense chemical imbalances, particularly the proton gradients found at the interface of different environments. These gradients acted like a natural battery, providing a steady flow of energy that a primitive metabolic network could harvest. This bioenergetic foundation provided the stability needed for more complex molecules like RNA to emerge and persist. To quantify how these systems transition from simple mixtures to persistent networks, researchers have developed the Thermodynamic Abiogenesis Likelihood Model, or TALM. This probabilistic model moves away from the idea of life as a lucky accident and instead treats it as a measurable outcome of energy regulation. TALM looks at variables such as the number of reactions in a chain, the energy thresholds required for each step, and the system's structural resilience. A key finding of this model is that selection-like behavior can emerge in any system where energy regulation enables sustained reaction cycles. This means that natural selection is not a property exclusive to biology, but a fundamental consequence of thermodynamics that can operate on purely chemical systems. It suggests that the path to life is paved by systems that are experts at staying in a state of dynamic stability, even when the energy coming from the environment is chaotic. This systemic perspective also helps solve the mystery of chirality, or handedness. Almost all biological molecules on Earth have a specific orientation. For example, the sugars in our DNA are all right-handed, while the amino acids in our proteins are left-handed. In a random chemical mixture, you would expect an equal number of both. A holistic view suggests that this wasn't a random choice made by a single molecule, but a systemic property that emerged as the network grew. Once a chemical system begins to favor one orientation through a process called symmetry breaking, that choice can be reinforced through autocatalytic feedback loops. In these loops, a molecule of a certain hand acts as a catalyst for the production of more molecules with that same orientation. Over time, this feedback leads to a frozen accident, where one orientation dominates the entire system. This is another example of how a property that seems like a fundamental rule of life actually emerged from the collective dynamics of a prebiotic system. By shifting our focus from the building blocks to the system as a whole, we begin to see the origin of life as a continuous process of increasing organization. We are no longer looking for a single eureka molecule. Instead, we are looking for the point where a disorganized mixture of chemicals undergoes a phase change, clicking into a highly ordered state where the parts begin to work in concert. This shift toward understanding life as a systemic phenomenon naturally leads us to consider the broader context in which these chemical networks operated. If life is the result of universal chemical and thermodynamic principles, then the environment that fostered it was not just a container, but a partner in the process. This raises the question of how these localized chemical symphonies began to harmonize with the global cycles of the planet itself. Chapter 3: Life as a Planetary Force. The search for life beyond our world has traditionally been a hunt for the individual. We imagine a rover scooping up a handful of Martian soil to find a single wriggling microbe, or a probe dipping into the subsurface ocean of an icy moon to capture a solitary swimming organism. But as our technological capabilities expand and our understanding of biology deepens, scientists are beginning to realize that life is not just a localized biological event. It is a planetary phenomenon. When life takes hold on a world, it does not remain a quiet, isolated passenger. It becomes a global force that fundamentally reshapes the planet's crust, its oceans, and most importantly, its atmosphere. This realization has transformed the way we look at the cosmos. Instead of searching for the needle in the haystack, the individual organism, we are learning to look at the haystack itself. We are looking for the smoking gun of a living world, a state of chemical disequilibrium and the presence of global biosignatures that can only be explained by the collective activity of a planet-wide biosphere. To understand life as a planetary phenomenon, we must first understand the concept of chemical equilibrium. A dead planet is a world in balance. Its atmosphere is the result of simple, predictable chemical reactions driven by heat, light, and geology. If you leave a mixture of chemicals alone for long enough, they eventually reach a state of rest where no further reactions occur. This is equilibrium. However, a living planet is a world in a constant state of restlessness. It is a world pushed far away from balance. On Earth, our atmosphere is a profound example of this. It contains high levels of oxygen and methane simultaneously. In a sterile environment, these two gases are highly reactive with each other. They should cancel each other out, reacting to form carbon dioxide and water. The fact that they both persist in our atmosphere in significant quantities is a biological miracle. It means that something on the surface is constantly, aggressively replenishing them. That something is the collective metabolism of Earth's life. Methane is pumped out by billions of microorganisms, while oxygen is released by the global machinery of photosynthesis. This massive departure from chemical equilibrium is perhaps the most reliable sign of life that a distant observer could detect. This shift in perspective from the individual to the planetary has given birth to the field of comparative exobiology. We are no longer just asking if there is life out there. We are asking how life begins and how it manifests on a global scale across different environments. To answer these questions, we are developing a new generation of eyes on the universe. While our previous efforts relied on detecting the shadows of planets as they cross the faces of their stars, a technique known as the transit method, we are now moving toward characterizing the atmospheres of these worlds directly. This is a higher technological hurdle. The light from a parent star is often 10 billion times brighter than the dim reflection from its planets. To see the planet, we must find a way to squelch the glare of the star. One of the most ambitious proposals to solve this problem is the star shade. Imagine a spacecraft shaped like a giant dark sunflower, the size of a baseball diamond, unfurling tens of thousands of miles ahead of a space telescope. By positioning itself perfectly, the star shade would cast a deep shadow over the telescope, blocking the starlight and allowing the instrument to capture the tiny, precious photons reflecting off a distant Earth-sized world. This would yield the first direct images of potential life-bearing worlds, not as detailed maps, but as small clusters of pixels. Yet even a handful of pixels can tell a profound story. By observing how the brightness of those pixels changes as the planet rotates, revealing the glint of an ocean or the different reflective properties of a continent, we can begin to reverse engineer the planet's albedo, its rotation rate, and even its seasonal variations. Once we can isolate the light from a planet, we can use a technique called transit spectroscopy to read its atmosphere like a barcode. As starlight passes through the gases surrounding a world, different molecules absorb specific wavelengths of light. When we split that light into a rainbow spectrum, the missing slices tell us exactly what is present in the air. A pattern of black lines might indicate the presence of water vapor, carbon dioxide, or methane. If we see oxygen and methane together, we have a strong argument for a living biosphere. We might even detect the red edge of photosynthesis, a specific reflective signature produced by the way plants or algae handle infrared light. This wouldn't just be evidence of a single organism, it would be evidence of a global energy harvesting system that has taken over the planet's surface. However, the search for these biosignatures is complicated by the fact that Earth has not always looked the same. Our planet is our only model for life, but it has been many different planets over its 4.5 billion-year history. Scientists refer to these as alternative Earths. For the first 2 billion years of its existence, Earth had a thriving biosphere of microorganisms, yet its atmosphere contained almost no detectable oxygen. An observer looking at Earth during the Archean eon would have seen a world with a tinted orange, methane-rich atmosphere, very different from the blue and white marble we know today. If we only look for oxygen, we might miss billions of years of biological history on other worlds. We must learn to recognize the signs of life in all its stages, from the early non-oxygen producing microbial mats to the complex oxygenated worlds of the present. This historical perspective is now being integrated into our mission designs. The upcoming Habitable Worlds Observatory is being conceptualized not just to catalog living worlds, but to test fundamental theories about how life begins. By surveying a large sample of at least 25 to 50 Earth-like planets, we can begin to look for patterns. Different theories make different predictions about the frequency of life. Some suggest that life is ultra-rare, requiring such a precise alignment of conditions that we might find no other biospheres within 33 light-years of Earth. Others suggest that life is an inevitable consequence of liquid water and alkaline hydrothermal vents, predicting that life will emerge readily wherever these basic conditions exist. By observing a large enough sample, we can move from speculation to statistical data. If we find even a single other biosphere, we falsify the theory that life is a vanishingly improbable accident. If we find none on 50 promising candidates, we support the rarity hypothesis. This statistical approach also applies to the environments that foster life's emergence. Recent studies using Bayesian analysis have revealed a surprising, counterintuitive possibility that fewer favorable environments might actually boost the chances of life taking hold. We often assume that more lottery tickets, more sites with the right conditions, would increase the odds. But the data suggests that when there are fewer sites, those sites are more likely to be of exceptionally high quality, perfectly conducive to the complex chemical transitions of abiogenesis. This could explain why life on Earth emerged so early despite a relatively harsh environment. It suggests that while our planet may not have been teeming with countless sites for life to start, the few sites that did exist, such as shallow volcanic pools or deep-sea vents, were extraordinarily favorable. This insight helps us target our search. Instead of casting a wide net, we might focus on finding high-quality environments that mirror the specific conditions of the early Earth. As we look for signs of biological life, we are also expanding our search to include technosignatures, the marks of a technologically advanced civilization. Just as microbial life alters a planet's atmosphere with oxygen and methane, an intelligent civilization might alter it with artificial pollutants or smog. We are looking for more than just radio signals, which have been the focus of the search for extraterrestrial intelligence for decades. We are now considering a much broader spectrum of evidence. This includes the detection of city lights on a planet's night side, or the presence of large artificial structures passing in front of a star. Even the energy use of a civilization would leave a trace. According to the laws of thermodynamics, any civilization that uses energy must emit waste heat, which would be detectable as infrared radiation. The search for technosignatures is fueled by the realization that our galaxy is teeming with planets. We now know that most stars host at least one planet, and that Earth-sized worlds in the habitable zone are common. This wealth of worlds has changed the calculus of the Great Silence, the Fermi paradox, which asks why, if life is likely to be plentiful, we haven't heard from anyone. Perhaps life is common, but the transition to a technological civilization is rare. Or perhaps civilizations are fleeting, appearing and disappearing in the blink of a cosmic eye. By searching for technosignatures alongside biosignatures, we are attempting to place our own history in a cosmic context. We are looking for anomalies in the data, irregular fluctuations in a star's brightness, or unexpected chemical signatures that might signal a presence far more advanced than our own. The challenge in all of this is distinguishing a true sign of life from a false positive. Methane can be produced by volcanoes as well as by microbes. Oxygen can be generated by the breakdown of water vapor by ultraviolet light. To be certain, we need more than a single gas. We need a holistic understanding of the planet's context. This means detecting not just the gases, but the planetary characteristics that support them. Is there an ocean to act as a solvent? Are there continental features to provide mineral diversity and wet-dry cycles? Is the host star stable, or does it erupt in sterilizing flares that would strip away an atmosphere? The detection of life will not be a single eureka moment, but a slow process of building a case piece by piece using computer models to simulate how these different factors interact. We are currently working on a framework to quantify this certainty, a scale that ranges from the first hint of a potential signal to the absolute certainty of a confirmed biosphere. This framework acknowledges that the road to discovery is winding. We might find a planet with a 95% probability of life, which would be a historic achievement even if we cannot visit it. As we peer deeper into the dark, we are looking for more than just another version of ourselves. We are looking for the universal principles that govern the transition from a planet of rocks and water to a planet of life and intelligence. We are learning that life is not a separate entity from the planet, but an integral part of its fabric, sustained by the same geological and atmospheric cycles that shape the world itself. This planetary view of life's origin and persistence suggests that the story of life is not just our story, but the story of the cosmos itself, written in the barcodes of starlight and the restless chemistry of distant atmospheres. This global interconnectedness, where a planet and its life form a single inseparable system, provides the necessary foundation for the emergence of even greater levels of complexity. Chapter 4. The Architecture of Interdependence. To truly grasp the origin of life, we must move beyond the idea of life as a collection of parts and begin to see it as a web of interconnectedness. This concept is the foundational rule of complexity. In the biological sciences, interconnectedness reflects the intrinsic reality that nothing exists in isolation. This web of interactions spans multiple layers, ranging from the intermolecular and interorganelle levels up to the intercellular, intertissue, and eventually the intersystem levels within a whole organism. When we look at the most complex systems we know, such as the developing human embryo or a functioning ecosystem, we see that the underlying processes are governed not by the individual components, but by the connectivity between them. This has led to the proposal of the in toto biological framework, which suggests that to understand how complexity emerges, we must observe the system in its entirety, shifting from an organ-centric view to a system-centric paradigm. The in toto approach is built on a triad of modern technologies that allow us to witness the birth of complexity in real time: organoids, intravital microscopy, and computer vision. Synthetic tissues, or organoids, display remarkable self-organizing properties, yet they often hit a developmental wall, remaining in an immature state. The reason for this stagnation is a lack of systemic connectivity. Without a circulatory system to deliver nutrients or hormonal signals to coordinate growth, the tissue cannot advance. By pairing these models with intravital microscopy and computer vision, researchers can track single-cell choreography, revealing that the instructions for complexity are distributed across the entire network of interactions rather than located in a single master switch. These same tools are now being applied to the study of protocell colonies, allowing researchers to watch the choreography of the very first chemical networks as they transition from random collisions to integrated systems. We find a powerful model for this transition in human fetal development, where the foundations of complexity are built with remarkable speed during the first 1,000 days, the critical window from conception to age 2. During the move from the first to the second trimester, there is an explosion of system-level interactions driven by the deployment of the circulatory system. Once the heart begins to pump, the state of being connected across distant tissues begins to dictate the behavior of every cell in the body. This period is not just a phase of physical assembly, but a crucible of long-term destiny. Through the concept of fetal programming, we see that a fetus's adaptations to its prenatal environment can permanently alter its physiology and long-term disease risk, demonstrating that the early dialogue between the developing system and its surroundings sets a trajectory that lasts a lifetime. The moment a system becomes interconnected is the moment it gains the capacity for higher-order function. One of the most striking examples of this interconnectedness is found in the relationship between blood vessels and the organs they serve. The in toto framework reveals that vessels are not just pipes but instructors. In the developing human liver, specific subsets of endothelial cells in the vessels produce angiocrine factors that tell liver cells how to proliferate and organize. A concrete example of this is the endothelial WNT2 hepatocytic FZD5 pathway, a species-specific interaction that guides human organ growth. This signaling is often activated by the physical force of blood flow itself, a process known as mechanotransduction, showing that the physical environment and the chemical signaling system are two sides of the same coin. This systemic instruction continues with the endocrine system, which acts as a global broadcast network. Hormones act as fundamental switches that trigger terminal differentiation, ensuring that the gastrointestinal tract, kidneys, and lungs all mature at a pace compatible with the rest of the system. In the context of life's origins, this suggests that the first chemical collectives must have had some way of broadcasting their internal state to their neighbors to ensure the survival of the whole. This reveals a nested arrangement of systems where the development of one entity is inextricably linked to the environment provided by another. The rules governing this nested complexity were originally proposed by sociologists to explain human development, but they are now being scaled down to the cellular and chemical levels through ecological systems theory. This perspective emphasizes that the primary engines of development are proximal processes, the enduring bidirectional forms of interaction in the immediate environment. It is not enough for the ingredients of life to be present. They must interact consistently over time. These interactions create reinforcing feedback loops that drive the system toward greater complexity. In this view, life is not a thing that was added to the Earth. It is a property that emerged from the enduring interactions of the Earth's nested systems. To understand this nested complexity, we can look at 5 distinct levels of interaction. The microsystem is the immediate environment, such as a cell's local chemical concentration. The mesosystem consists of the connections between different microsystems. The exosystem involves indirect influences, such as distant geological shifts that alter local acidity. The macrosystem is the broad context, like the overall chemistry of the early Earth. Finally, the chronosystem is the dimension of time, encompassing the historical cycles that reshape the system across its lifespan. The necessity of interconnectedness is also reflected in the thermodynamic requirements of persistent networks, where a viability inequality must be maintained. For a complex system to survive, it must use its interconnected subsystems to balance energy input and expenditure while buffering against environmental change. This systemic view even helps us understand the evolution of the brain and its connection to the rest of the body. The foundations of cognitive ability are built early and are deeply intertwined with the signals the brain receives from the immune system, the gut, and the circulatory system. Technical metrics of this interconnectedness show that a healthy developing brain is characterized by a shorter average path length for efficient structural communication and a higher between the centrality for the nodes that play integrative roles in information flow. As we look at the history of life on Earth, we see a pattern of biological leaps in integration, where the benefits of cooperation consistently outweighed the costs of independence. The emergence of complexity is a middle-out process of enduring interactions where a chemical network learns how to manage its own energy and information through the use of its neighbors. This systemic intelligence is what allowed life to not only originate, but to persist for billions of years, adapting to every catastrophe the Earth could throw at it. This shift toward a system-centric paradigm has profound implications for how we search for life on other worlds. If we accept that life is a property of interconnected systems, we must look for environments that can support those connections. We are looking for a living system rather than just a living thing. This planetary perspective suggests that the story of our own origins is not a unique, isolated event, but a specific instance of a universal process where matter, energy, and information become inextricably linked. This universal evolution provides the ultimate context for understanding how the transition from non-living chemicals to complex thinking beings was possible. If the same rules of nested systems and proximal processes apply across the cosmos, then the signature of life may be written in the very way a planet integrates its disparate parts into a functioning whole. The struggle to define what we are is perhaps the oldest intellectual hurdle in human history. In our search for the origins of life, we have almost always started with the assumption that life is something possessed by an individual. We see a bird, a tree, or a bacterium, and we say that this specific entity is alive. However, as we push the boundaries of astrobiology and look toward the stars, this individual-centric definition is being fundamentally challenged. Scientists are finding that our traditional ways of categorizing life are not just incomplete, they may be actively hindering our ability to find life elsewhere. By shifting our perspective through the lens of comparative exobiology and the concept of universal evolution, we are beginning to see life not as a collection of separate objects, but as a continuous, multi-scale process of matter and energy organization. The difficulty begins with the very word life. There is currently no single, universally accepted definition that satisfies every scientist. In fact, a survey of the scientific literature reveals at least 123 different published definitions. Some focus on metabolism, others on physiology, biochemistry, or genetics. One of the most famous is the working definition used by NASA, which describes life as a self-sustaining chemical system capable of Darwinian evolution. While this is a useful practical guide for designing missions, it contains a startling paradox. According to this definition, a single individual, like a lone rabbit or even a human being, is not technically alive, because an individual cannot undergo Darwinian evolution on its own. Evolution is a property of a population over generations. This realization forces us to acknowledge that the individual is not the primary unit of life. The lineage or the system is. This challenge to the individual is even more apparent when we look at the biological concept of units of selection. In many colonial organisms, the line between an individual and a group disappears. Consider a colony of genetically identical organisms that reproduce asexually. In biology, the entire colony is referred to as a genette, while the individual members are called ramets. In this scenario, it is the genette, the collective, that functions as the unit of selection in the eyes of evolution. The survival of the group is what matters, and the individual members are almost like the cells of a larger body. If we apply this logic to the origin of life, it suggests that the first living systems were not solitary first cells, but were likely vast interconnected networks or microbial mats where horizontal gene transfer was so rampant that the concept of a distinct species or individual had no meaning. This disciplinary rift in how we define life is visible in the very way we plan our scientific future. A fascinating detail emerges when comparing major institutional reports. The Astro 2020 Inches Decadal Report, which sets the agenda for astronomy and astrophysics, never once mentions the term origin of life. In contrast, the Origins, Worlds, and Life (AWL) report treats it as a central defining pillar. This highlights a profound gap in how different fields categorize the phenomenon. To an astronomer, life might be a planetary signature. To a biologist, it is a specific chemical history. Bridging this gap requires a definition that is entirely molecule agnostic. From the perspective of physics and thermodynamics, an organism is not an object, but a thermodynamic system with an organized molecular structure that makes use of energy gradients to create copies of itself. This view treats life as a self-organizing and autopoietic process, a system that produces itself. This shift from what life is made of to what life does is a hallmark of universal evolution, which treats life as a specific stage in the increasing complexity of matter. It also forces us to confront the fundamental debate of how life's engine first started, the conflict between autotrophy and heterotrophy. Did life begin by creating its own food from inorganic sources, or did it emerge by scavenging existing geological molecules? This is not just a question of ancient history. It is a question of whether life is a self-starting planetary imperative or a lucky consumer of a pre-existing chemical buffet. This broader view is essential for the field of comparative exobiology. To break out of Terran chauvinism, we are looking for a second genesis, an independent origin of life. This search is driving the development of the Large Interferometer for Exoplanets, LIFE, a critical future facility designed specifically to constrain the conditions necessary for the origins of life on other worlds. Alongside other observatories, LIFE will help us determine if the chemical pathways we see on Earth are universal constants or local accidents. The search for this second genesis also drives us to look for signs of life that have nothing to do with individual organisms. When we detect a planetary atmosphere in extreme chemical disequilibrium, such as the reactive coexistence of oxygen and methane, we are seeing the signature of a biosphere as a single functioning unit. Similarly, the search for technosignatures, the collective footprint of intelligence, looks for signals that transcend the biological individual. One of the most promising hailing channels for such a search is the hydrogen line at 1,420 MHz. Because neutral hydrogen is the most abundant element in the universe and emits radiation at this specific universal frequency, it serves as a natural constant. Any advanced civilization, regardless of its specific chemistry, would recognize this frequency as a universal landmark, making it the logical choice for a cosmic greeting. This idea of collective intelligence may actually be the key to understanding our own cognitive evolution. A new theory called complementary cognition proposes that humans did not evolve to be individually brilliant, but to be collectively adaptive. According to this view, our ancestors evolved to specialize in different but complementary ways of thinking to navigate a highly variable environment. For example, neurocognitive specializations like dyslexia, which are often viewed as disorders in a standardized educational system, may actually be vital components of a group-level search strategy. One individual might excel at exploiting known solutions, while another, the individual with dyslexia, is specialized for exploring new, innovative possibilities and global pattern recognition. In this framework, the human mind is a distributed system, where the unit of success is the group. Language then evolved not just for communication, but as an inheritance mechanism to share the results of this collective cognitive search. This systemic perspective also changes how we view the probability of life's emergence. By applying statistical analysis to the trajectory of civilizations, researchers are finding that the transition from chemistry to biology and from biology to technology may follow a continuous and perhaps predictable path. If the conditions are right, the emergence of a collective intelligence that can manipulate its environment and broadcast its presence to the stars might be an inevitable consequence of universal evolution. This perspective offers a potential explanation for the great silence of the Fermi paradox, suggesting our definitions may simply be too narrow. Even the history of our own planet supports this move away from the individual. For the vast majority of Earth's history, the world was dominated by microorganisms. Bacteria have been the primary drivers of the planet's ecological circuitry for billions of years. From a planetary perspective, multicellular organisms like humans are a very recent and perhaps optional icing on a bacterial cake. The heavy lifting of maintaining the Earth's habitability is done by the collective metabolism of the microbial world. This suggests that if we find life elsewhere, it is far more likely to be a global microbial system than a collection of distinct, large individuals. This shift toward a planetary and universal view of life is reflected in the way we are designing our future telescopes, to move from the study of the individual planet to the study of planetary populations. By observing how life-bearing potential scales across different environments, we can begin to see the choreography of universal evolution on a galactic scale. Ultimately, challenging the individual-centric definition of life allows us to see ourselves as part of a much larger story. We are not just isolated beings living on a rock. We are a manifestation of a universal process that began with the first stars and continues through the collective intelligence of our species. This perspective trivializes the differences between us and emphasizes our shared identity as Earthlings, the inhabitants of a single living world. It also prepares us for what we might find in the dark. If we encounter a system that is self-organizing, autopoietic, and capable of managing energy and information, we will know it is alive, even if it doesn't have a single cell or a strand of DNA. The origin of life is not a mystery of how a single thing began, but how a universal process of interconnectedness took hold on a small blue marble and began its long journey toward the stars. As we prepare to take the next step, we are armed with a new framework that recognizes life as a planetary phenomenon, a collective effort that bridges the gap between the smallest molecules and the vastest reaches of space. This understanding doesn't just change how we look for life elsewhere, it changes how we see ourselves and our place in the unfolding history of the universe. The quest to find our origins by looking beyond the individual is leading us to a future where the discovery of a second example will finally allow us to move from the study of Earth's specific history to the mastery of the universal laws of biology. What will those laws reveal about the ultimate limits of complexity in a universe that seems designed to produce it? Our exploration reveals a profound shift in perspective. Life is not a solitary accident, but a collective masterpiece. By moving beyond the individual-centric view, we see that the transition from chemistry to biology was driven by systemic intelligence and planetary-scale cooperation. Life is an emergent property of the universe, a continuous process of increasing complexity linking the smallest molecules to the vastest reaches of space. This holistic framework suggests that the smoking gun for life elsewhere may not be a single microbe, but a world in restless chemical disequilibrium. As we search for a second Genesis, we realize we are not just observers of the cosmos, but a manifestation of its inherent drive toward interconnectedness. If life is a planetary imperative, how many other worlds are currently singing their own chemical symphonies? Thank you for listening.